Hello and welcome to Know All for GCSE. In this video, we will be looking at every single exam question for organisation, which is the second topic of the GCSE biology spec. Now, in this video, you'll find at least 3 exam questions on each subtopic in organisation. I've compiled a massive document and we'll be going through the answers. You can find the document itself in the description and in the comments pinned at the top, where you can download it for free and answer along with the video. So we're going to begin with the first subtopic, principles of organisation. Each of the subtopics will be time-stamped, and each of these resources have come from all of the papers, from the specimen papers to 2018 to 2022, along with textbooks. So this is the only exam question resource you will need for your exams. So the first question, describe the relationship between cells, genes, DNA, and chromosomes. So this is definitely a question where you're going to have to use your knowledge, and mainly your knowledge. There's not much understanding to it. You just need to know the hierarchical structure. So let's look at the answer. The cell contains a nucleus. The nucleus contains chromosomes, and chromosomes are made up of coiled strands of DNA. A gene is a section of DNA that codes for a protein. So in that, you've discussed the relation going from the biggest thing, the nucleus, which contains chromosomes. Chromosomes are made of DNA, and a gene is a part of the DNA. Okay, the next subtopic is the digestive system, but there's also some questions about stem cells, which is the B1 topic. So the first question is, explain the enzymes needed in digestion. Now, because this is 4 marks, you might have to talk about what each enzyme does as well. So in this case, oh, I'm so sorry, this is supposed to say explain why enzymes are needed. My apologies for that. You'll probably see the change in the document. So explain why enzymes are needed in digestion. So enzymes are biological catalysts. They break down large insoluble molecules into small soluble molecules that can be absorbed by the blood. So they're basically making big food bits into small bits so that our body can actually use all of the minerals from them. So biological catalysts break down of large insoluble molecules to small soluble ones that can then be absorbed by the blood. Explain how the release of bile into the small intestine increases the rate of fat breakdown. This is a 3 marker. So in this question, you need to use your knowledge of bile and its role in the digestive system, where it comes from. So the role of bile, it contains 2 roles. Firstly, to neutralize the acid, because the bile itself is alkaline, and then it hence helps to maintain the optimum pH for lipase enzymes, because as you know, fats, which you're being asked about in this question, are known as lipids, and the things that break down these lipids are lipase enzymes. And therefore, by making it the optimum pH, by neutralizing the acid, you're helping the lipase enzymes. And the second role of the bile is to emulsify fats. Now, what emulsifying means is basically cutting it down from big molecules into smaller molecules. And this helps to increase the surface area to volume ratio. And therefore, that because there are more surfaces for the enzyme to act on, it can be broken down much more quicker. The next question, explain why protease works more effectively in the stomach. This is again a 3 marker. So in this question, you have to use your knowledge of, again, conditions, rates of enzymes. Now, enzymes tends to come up a lot in exam papers, so make sure you're really familiar with these questions. So the answer is the stomach contains acid. Acid has a low pH, and this then provides optimum conditions for the enzyme protease to work. Now, this next question is a specialized question, that's why there's a star, so it's more difficult and it's relating to a specific context. So, in a person with cystic fibrosis, cells lining the lungs and the digestive system create too much mucus. Explain why children with cystic fibrosis grow more slowly than children without cystic fibrosis. Now, this was actually a question found in paper 2. However, I thought it was relevant in terms of the enzymes and that portion of it. So if you're only strictly revising just organization, you do not have to answer this topic. But if you are revising the whole of biology, then I would recommend you look at this, or it just gives you a greater understanding. So it's kind of connecting together all of the biology knowledge. You've got what happens in the pancreas. So the pancreas, there's fewer digestive enzymes which enter the small intestine, so enzymes break down less food, because remember, enzymes, there's pancreatic juices, which are the different enzymes. So because there's so much mucus, it lessens the amount of enzymes that can enter the small intestine, and therefore there's less absorption of nutrients, there's fewer amino acids, and so on, and going into paper 2 content. Okay, I'm just going to skip those 2 questions. I'll show you the answers if you did want to answer them. However, they're not strictly relevant to paper 1. So let's go on to. So moving on to the next subtopic, which is all about the food tests required practical. So as I said in my previous video, for all the required practicals, I've written you 6-mark responses, and if you put every bullet point that is listed on there, you will no doubtedly get 4 marks in any question about it. So what I want you to do is just mind map everything you can remember about the food tests, and I'm going to reveal the answers now. Okay, let me just make sure there's some on the other page as well. Okay, so let's go through the food tests required practical. So firstly, what you're going to do, and this is part of, it's contradictory whether it's part of the A level, do you need to mention this or not, but I would say mention it to be safe. So use a pestle and mortar to break up the food into small particles. That is the A level / GCSE bit, but if you do include it, I guess the examiner will only be impressed. Add this crushed food into a test tube with water and mention a specific volume. Shake to get these chemicals to dissolve. If testing for fats, use ethanol instead of water, because why? Fats do not dissolve in water. Then you're going to remove any undissolved solid material through filtration. And then you've got the following tests. So for the tests, you've got starch, which is you using iodine solution, and if starch is present, it will turn dark blue or black. Protein, which you use burette solution, and it should turn violet. And for tests for fats and sugars, so for the fats, it's a Sudan regiment test, and you get a red layer at the top. And for sugar, you do the Benedict solution test and sugar, and you would put it in hot water. Always make sure when you're talking about the sugar test of Benedict solution, you must mention, otherwise you won't get the mark, that you need to put it into hot water. And then it should turn orange. Okay, moving on to the next subtopic, which is about the rates of enzymes. So similarly to what we were just discussing. So the first question is the effect of pHs on enzymes required practical. So this is the second required practical that's closely related. So let's look at the answer for this. So the buffer solution, you're going to have a buffer solution, and the purpose of this buffer solution is just to contain a constant pH value. Often it's pH 5. You're going to put 1 drop of iodine into each spotting tile. You're going to take 3 test tubes and put 2 centimeters cubed of starch, 2 centimeters cubed of amylase. So amylase is the enzyme that breaks down starch, and 2 centimeters cubed of the pH 5 buffer solution. And that's to ensure that there's a constant pH. You're going to put all 3 test tubes in a water bath at 30 degrees. So again, that is a variable that you want to keep the same. And you're going to do that for 10 minutes. You're going to transfer 1 drop of solution into a spotting tile well every 30 seconds until the iodine remains orange. So what you're doing is you're seeing how long it takes for the starch to be fully broken down by the amylase, because the iodine will remain orange when it detects no more starch because it's been broken down. I hope that makes sense to you. If not, I would recommend going to watch Free Science Lessons video on that topic. It's really helpful. So the next question, describe the effect protease would have on baby food. Now protease, in case you didn't remember, is the enzyme that breaks down proteins. So let's see the answer. So just think, in this context, a lot of students got this question wrong because it came in an exam paper. Think about what's being asked. If you're unsure what to answer, think about the context given. Baby food. Now, obviously, babies can't have really crunchy material to eat because of their lack of development in teeth. So they need soft and runny food. And what does protease do? Protease breaks down the food, specifically the proteins, into amino acids. And remember, proteins are long chains and amino acids are slightly shorter chains. And therefore, by making it shorter, you're making it softer and runnier and easier for the baby. So that's a thinking outside of the box type of question. Okay, next question. Describe how ribosomes and mitochondria help to make enzymes. This is a 3-marker. So the ribosomes synthesize, which means to make. So ribosomes make proteins and enzymes using the amino acids, and then the mitochondria releases energy for the process. If you're ever asked how does mitochondria help in any process, it's always along the lines of the mitochondria undergoes respiration in order to release energy for work to be done. The next question, explain the lock and key theory of enzyme action. So this question comes up a lot each year, just in different forms. So here's one answer you could say. The enzyme's active site binds to the substrate because they are complementary. So keywords that the examiner will be looking for: enzyme, active site, substrate, and complementary. Hence, the substrate is broken down into products, and the products are then released, and the enzyme itself is not changed. Now, sometimes this particular clause at the end, the examiners will say that that's a very important clause, that you cannot get the 3rd mark unless you mention that the enzyme has not changed, just the product has changed, the substrate has changed. Okay, so here is again a more context question, and you can see the different questions here. So instead of going through this, I'm just going to leave the answers up on the screen so that you can just pause the video and use them at your leisure, but they're not necessarily in need just for understanding the topic. So the next question is, explain why amylase does not work at 5 degrees Celsius and 80 degrees Celsius. So this is a 5-marker. So you've got to think quite methodically when answering this question. You're given a temperature, really a low temperature and a really high temperature. Now normally this would be accompanied with a graph, but if you know enough about enzymes, you wouldn't need the graph, because you know that 5 degrees is too cold for the enzyme to work. It needs to meet its minimum activation energy, and 80 degrees will mean that the enzyme will denature. Now you've just got to put your ideas into a form that will achieve 5 marks. So let's look at that. At 5 degrees Celsius, amylase has a low kinetic energy and therefore fewer enzyme substrate collisions, and hence it works less and does not break down as much food. At 80 degrees, the amylase enzyme denatures and its active site changes. And if you wanted to go specifically into that, the reason why the active site changes is because the bonds holding together the shape of the active site change and break. The next question, what is an enzyme? So hopefully you would have answered this question long enough to know. An enzyme is a chemical which catalyzes reactions in living organisms. This is only 1 mark, so a simple answer like this would be fine. But if it was a multi-mark question, then I would definitely recommend going into more about how enzymes work. And the final question for this topic: explain how pancreatic cancer may cause a person to lose weight. Now, cancer is a topic that comes a bit later in biology, but it's still worth going through this because it talks about digestion. Food is not fully digested and less glucose absorbed into the bloodstream. So, because of, remember, we said if there's any problem with the pancreas, you know that there's going to be problems, metabolic issues, because the pancreas produces all of the enzymes. And because there's pancreatic cancer, you're not releasing enough enzymes, and therefore your food is not digesting enough. And therefore, you've got less glucose. When there's less glucose available, that means that less respiration has to occur because respiration gives energy from glucose, and therefore you will use more fat instead. Now, there will be fewer amino acids absorbed into the blood, and therefore fewer acids making protein for growth and repair. And let's just finish that answer. Therefore, there are fewer fatty acids absorbed, so less fat is actually stored in the body. Okay, moving on to the next subtopic, which is the heart, the blood, and blood vessels. So the first one is a 6 marker. Describe the levels of organization in the human circulatory system. So take a deep think about this. Think about how it's a double circulatory system, so how would both paths work? And I'm now going to reveal the answer. So muscle cells can contract and respiration occurs in the muscle cells. The muscle cardiac tissue then work together to contract and respire, and as a cardiac muscle does not fatigue, the heart, which is the organ, the muscle valves, and the blood vessels all put the blood under pressure. And the circulatory system is made up of the heart, the blood vessels, all working together to pump blood around the body. Now, that is a different method of attempting this question. A lot of students thought, let's discuss both circulatory system, and in this case, it is a really tricky way to tell. Hopefully, you don't get a really vague question like this where you're unsure which path to go. The next question: explain why the left side of the heart is thicker. Now, do note that in future exams they won't ask this question again, but they will mask this question under another situation. So be very careful and remember that the left side is thicker, and the exact reason why. Let's read it. So the left ventricle pumps oxygen around the entire body, but the right ventricle only needs to pump blood to the lungs in order to go and get oxygen. So if you've got one thing pumping to the whole body and one only to the lungs, then the left ventricle needs to generate a much greater force of contraction, and therefore they need thicker walls to withstand this greater pressure. Okay, let's move on to the next question. Compare the structure of an artery with the structure of a vein. So this is a lot of your knowledge and comparison techniques. So arteries have a thicker layer of muscle than veins, and arteries have a narrower lumen. So a lumen is practically just the hole, and that allows blood to go through. So arteries are much more narrower, whereas veins are thinner and wider. Veins also have valves, but arteries do not have valves. And arteries have more or thicker elastic tissue. The next question: define the term double circulatory system. This is only 1 mark, so you do not need to put too much attention into this. So a double circulatory system is when there's blood that's being pumped to the lungs on one side of the heart and to the body by the other. Now, 6 marker. Explain how the human circulatory system is adapted to supply oxygen to the tissues and remove waste product from the tissues. So this is asking you to be specific in terms of the adaptations into how is it adapted so that it can get a lot of oxygen around the body and also remove the waste products. Okay, so we've got quite a hearty answer here. So, the double circulatory system allows for higher blood pressure and greater flow of blood, because imagine if there's one path, there will be a lot of people on that one path, so it'll be more congested. But if there were two paths, this would make it much easier to move around, and therefore it would allow a greater flow of people or a greater flow of blood to be supplied to the tissues. Another adaptation is that the pulmonary artery carries the deoxygenated blood to the alveoli, and the alveoli is where it absorbs oxygen, and it's got a thin wall, elastic tissue, discuss all of that. This is carried back to the heart in the pulmonary vein before being pumped to tissues by the heart through the aorta. Oxygen is carried by red blood cells, which contain no nucleus, and the whole point of blood cells containing no nucleus is so that it can hold more hemoglobin for oxygen transport. And remember, hemoglobin binds to oxygen to create oxyhemoglobin, which is the thing that makes the red blood cells red. The capillaries carry blood into the tissues. They have thin walls to allow for optimum diffusion of oxygen out and waste products in. It also has a higher surface area of capillaries for optimum diffusion, and the blood goes back to the heart in the veins with the valves to prevent backflow. So we've discussed numerous adaptations. You definitely do not need to talk about all of them in a single 6 marker, but the more you talk about, the stronger your chance and ability of getting top grade is. Okay, the next question and the last question of this subtopic. Explain why having only one ventricle makes the circulatory system less efficient than having two ventricles. So what you need to think about here is what is the function of ventricles, and therefore how would not having one prohibit and limit the body. So, in the mark scheme it detailed, oxygenated and deoxygenated blood would mix if there was one ventricle. And if it would mix, that means that there's actually less oxygen that reaches the body cells. Because if you're thinking about diffusion or transport of gases, you need to know that it needs to maintain a steep concentration gradient, which just means that there's a difference. There's an area of high oxygen and then there's an area of low oxygen so that it can diffuse quickly. However, if it's all mixed up together, then it will not really have a concentration gradient, so oxygen would not move across. The next subtopic is ventilation and gas exchange. This is a little smaller one, yet it still is a 6 marker. So explain how the human lungs are adapted for efficient exchanges of gas via diffusion. So if you want some tips, it will be talking about alveoli, bronchi, the rings of cartilage, etc. So there's many alveoli, which increases the surface area to volume ratio. The capillaries mean that there's a short diffusion path for oxygen. The breathing mechanism and the diaphragm moves air in and out subconsciously, which allows you to bring in fresh oxygen and remove all that waste product of carbon dioxide. And also talking again about the capillaries, there's a large capillary network, which means that there's a good blood supply to rapidly bring in oxygen and to rapidly move out carbon dioxide. On to the next subtopic. Explain why a stent prevents a heart attack from happening. So remember, you have statins and you have stents. So let's see how this works. So this is 3 marks, and what's mentioned in the mark scheme. A stent keeps the coronary heart open, because remember, it's that mesh coil. So it keeps the coronary heart open so that blood flow can carry all those needed things in the body. So all that needed glucose and oxygen, and therefore glucose and oxygen is needed for respiration. Okay, the next subtopic, non-communicable diseases. List the risk factors of coronary heart disease. So remember, risk factors are things that can lead to whatever disease you're talking about. So what things can lead to you having heart problems? Well, lack of exercise, obesity, smoking, stress, alcohol consumption, because that reduces the capacity and abilities of things like your lungs and your liver, your diet, and your sugar. This next one is a 6 marker. So this is evaluating the use of cholesterol blockers versus statins for a person with high cholesterol. Now, I remember specifically in this question, you were actually given some context about cholesterol blockers, so it's unfair for you to just be expected to know it now. So I'm just going to give you the answer here, and you can check this in your own leisure. Although you should know about statins because that's in the syllabus. Now, what condition may be treated using an artificial pacemaker? Now, through this question, it's testing, do you know what a pacemaker does, and hence what condition is there which is caused by you not having a good one? Let's look at the answer. Now, this is a irregular heartbeat, or known as chorusarrhythmia. Now, the pacemaker controls the heartbeat, and I think it's found in the right ventricle, and it has these small electrical impulses that cause your whole heart to contract, and that's known as your heartbeat. So that's why you would need an artificial pacemaker to treat something like if you had an irregular one. This next question is comparing mechanical valves to biological valves. So firstly, make sure you know what valves are and mention them, and then talk about mechanical versus biological. So mechanical valves are more durable and longer lasting than biological. So an example of a biological valve could be a valve from a pig, for example. And hence it's less durable than a mechanical one. But mechanical valves can also cause blood clots to the brain, which could be really dangerous. And this means that the patients need to take anti-clotting medicine in order to prevent that from happening. And medication can lead to excessive bleeding after the injury. However, there is still a higher survival rate with mechanical, and you would be told that in the context. With biological, there is no additional medical requirements. However, there could be some ethical issues because it's an animal you're taking it from, and the valve may be harder. You're more likely to need a replacement valve with biological valves, and it's more likely to be rejected because your body only rejects things that have entered your body that are living. For example, a virus. It wouldn't reject a mechanical valve because that's made of metal, which is not living. Next question. Explain why a person with a leaking heart valve has difficulty exercising. So again, use your knowledge on what valves are, what their function is, to prevent the backflow of blood, and how would that then mean that if there was an issue, how would it cause you to have difficulty in exercising? So this is again a methodological one where you have to go from one step to another. So because backflow of blood can occur, this means that less blood is pumped around the body efficiently, so that means less oxygen is applied to the muscles, therefore less aerobic respiration takes place, less energy is released, less efficient muscle contraction, and also your body has to rely therefore on anaerobic respiration, so respiration without the need of oxygen. But this causes an oxygen debt which eventually needs to be paid, and also muscle tension because of the lactic acid. The next subtopic, the effect of lifestyle on non-communicable diseases. So this 6 marker is asks to explain the risks of a pregnant woman smoking and drinking. So here in the mark scheme, they've discussed both topics separately. So for smoking, they've said that there's carcinogens, which increase the risk of cancer. Carbon monoxide is a toxic gas, and carbon monoxide is really bad because it reduces the amount of oxygen that your red blood cells can hold, and this means that it can cause fatigue, and you're less likely to create energy and muscle fatigue. And for the fetus, this means that because it can't get enough oxygen, it could actually suffocate. This increases the risk of having miscarriages, being premature, and SIDS as well. Alcohol. In the first 3 months, having alcohol can actually increase the risk of having a child that's premature or having a miscarriage. And heavy drinking that's sustained can actually lead to FAS, which is fetal alcohol syndrome. The next question. Describe the effects of liver failure on the human body. So, for this question, think about what the purpose of liver is, what does it do, and therefore what would its absence do to the body? So, the liver, one of the functions of the liver is to produce bile. So, if there was liver failure, it wouldn't produce bile, and therefore the fats and the lipids would not emulsify, and the pH of the small intestine would not be neutralized, which means that the enzymes like lipase would not work effectively. The food would not be digested properly. This means that a person may lose weight. Lactic acid may not be properly broken down or oxidized. The accumulation of lactic acid is toxic and it could actually cause body poisoning. Proteins, such as amino acids, won't be broken down, so there would be no deamination, and therefore ammonia would form, which again is toxic, and glycogen stores would not also be formed. Now, this is kind of a mix up of paper 2 content as well, so do forgive yourself if you didn't get all of those marks there, and make sure that you remember it's paper 2 content as well. So the next question is explain how diabetes can cause the body to lose more water. So when someone has diabetes, it actually means that the blood is more concentrated than the solution within the body cells. Therefore, water moves out of the cells via osmosis through a partially permeable membrane. And because more water is moving out, you're more likely to have more water being wasted and lost. Suggest 2 reasons why data isn't considered valid, and suggest 1 factor that might reduce the validity. This is just practical questions that I thought might be helpful, so I added at the bottom. So a large number in the survey, or a well-controlled survey, and an under or over estimate of alcohol consumption, people's lifestyles may change over time. So the next subtopic is cancer. Compare the similarities and differences between benign and malignant tumours. So, similarities include that both are uncontrollable growths. Both are formed from a person's own cells. However, differences start now. Benign tumours are enclosed and in a barrier by a membrane, so it can't leave the membrane. And therefore, because it can't leave, it doesn't invade nearby tissue. So that's good for humans, because if a tumour is not allowed to go and invade the rest of your body, that means it's just limited to that one area. Whereas with malignant tumours, these are not enclosed and they are therefore cancerous. They can spread and invade and form secondary tumours around your body. Name some risk factors of cancer. So we already discussed what is a risk factor. Anything that causes something to happen. So HPV linked, the carcinogens in smoking, alcohol intake, UV radiation, diet, all of these things have links to the ability to get cancer. Smoking can cause diseases such as cancer. Now explain why cancer is not a communicable disease, which just means a disease that can spread. So you need to realize that cancers caused by smoking cannot be passed from person to person, and the cancer itself is not caused by a pathogen. Communicable diseases tend to be caused by pathogens and other living organisms, whereas cancers tend to be caused by a person's own body cells. Now, Helicobacter pylori, or acid tolerant bacteria, can damage the human mucus lining. Suggest how an infection might result in a stomach ulcer developing. So, bacteria are not killed by the hydrochloric acid because they are acid tolerant, and then that means that they can damage the mucus lining, and therefore the hydrochloric acid would cause an ulcer because it's therefore getting into contact with other things that is behind the mucus lining. And finally, describe how a person infected with Helicobacter pylori could also develop liver cancer. And you would say that if the cancer is malignant, which means it spreads, that means that they can spread to other organs and they move via the blood, forming secondary tumours. Okay, let's move on to the next subtopic, which is the leaf structure. So, the changes in the mean width of the stomata in normal conditions are an advantage to the plant. So, what they're asking you here is to talk about how does the plant itself, how is it able to change the size of the stomata, and how is this a benefit to the plant? So, let's look at the answer here. The stomata are closed at night because there is less or very little photosynthesis, because remember it has to occur in sunlight. Closing this stomata reduces and prevents water loss. Stomata then opens wide at midday as maximum light intensity for the photosynthesis occurs. And the stomata opening wide to take in more carbon dioxide also does this for photosynthesis. The stomata, yeah. Moving on to the next subtopic, which is all about transpiration and translocation. Well done for getting this far. We are drawing to a close in this video. So define and explain the process of transpiration. So transpiration is the movement of water and mineral ions, don't forget that, from the roots to the leaves via xylem tissue. Evaporation in the leaf of water from inside the cells. It diffuses through the air spaces and out through the stomata. Water passes from the xylem to the leaf to replace that lost water. Water is then drawn from the root hair cells to the xylem cells. And that's how the transpiration works. The next exam question is to compare the process of transpiration to translocation. So, in transpiration, it uses the xylem tissue, and this transports water from roots to the leaves. It's a one-directional flow. The movement of water leaving in evaporation causes a transpirational pull, drawing out more and more water from the roots. Whereas in translocation, the phloem tissue transports dissolved sugars from the leaves to the rest of the plant. So it's a multi-directional flow. And it also has sieve plates which help to control the flow. The phloem tissue is not dead, unlike xylem, and both are essential for the plant to survive and grow. Now, the next question, describe 3 reasons why transpiration is important, and this is the final question. You guys almost made it. Now, the 3 reasons are, firstly, it transports dissolved mineral ions, which are key for plant growth. Secondly, water is required for photosynthesis to work, and therefore all of the reactions to make food and energy. And finally, the evaporation of the water helps to cool down the plant, especially in warm conditions. Well done for getting all the way through the video, and I hope you found this video helpful. I'll be releasing these videos for biology, chemistry, and physics that will be coming soon. Again, thank you so much for watching, and see you soon.